Welcome back to the deep dive. So today we are going to challenge a pretty central piece of biological dogma,
A big one, yeah.
the spliceosome. That giant molecular machine that cuts and pastes our mRNA.
Right, the editor-in-chief.
We've always been taught it's the only game in town, but a new paper from the Elledge and Kennedy lab suggests, well, that's not the whole story.
It turns out the cell has a secret backup system, a kind of shadow editor.
And it's called SOS splicing.
Exactly. It's a mechanism that surgically removes parasitic DNA, these transposons, directly from the mRNA to rescue a gene. And it does this completely on its own. No spliceosome required.
That's huge, and this is all about dealing with transposons, these so-called jumping genes, right?
Yeah, the ones that copy and paste themselves all over the genome, they make up, what, almost half of our DNA?
And usually the cell just tries to shut them down at the DNA level, lock them up.
Right, but sometimes they escape. And if one lands right in the middle of an important gene, that's usually curtains for that gene's mRNA.
It's like someone pasting a random garbled paragraph into the middle of a sentence.
And we thought the cell just had to live with that, because the standard spliceosome, it just can't fix that kind of insertion.
Okay, so SOS splicing is the fix. But that raises the big question, the one in the first figure. How does the cell even know a transposon is there? How does it spot the bad RNA?
Well, it's all about geometry. The transposons they looked at have these sequences at their ends called inverted terminal repeats, or ITRs.
So the end of the sequence is basically a mirror image of the beginning.
Precisely. And when that gets transcribed into mRNA, those two ends, they just snap together like magnets.
And that forms a hairpin loop, a physical kink in the RNA strand.
Yes, and that hairpin structure,
Right.
that's the signal. It's like folding the corner of a page in a book to mark an error. That shape becomes a beacon for the repair crew.
I see. So the cell isn't reading the sequence, it's feeling the shape.
Exactly. And that beacon recruits a very specific surgical team, which brings us to the second figure. And this is not the spliceosome's crew.
It's a specialized trio of proteins.
Right. First, you have the scout, a protein called AKAP17A. It's the one that spots that hairpin error.
Okay, so it binds the target.
Then you have a bridge protein, CAAP1, which sort of holds everything together.
And finally, the heavy hitter, RTCB.
That's the RNA ligase.
The molecular glue that seals the two ends of the mRNA back together after the transposon is cut out.
Scout, bridge, and glue. A completely independent editing suite.
So here's a thought for you. This entire mechanism, it was first found in C. elegans, a tiny little worm.
Mm-hmm.
Now, when you hear that, it's easy to think it might just be some, evolutionary quirk, a niche thing.
Sure.
But do you think this specific machinery really survived hundreds of millions of years of evolution to make it all the way to humans?
Well, that's the kicker. Yes.
Yeah.
It's absolutely conserved, from worms all the way to us.
Wow. So that implies this is an ancient and really essential defense system.
Evolution decided it was too important to lose. The authors even proposed this two-tier defense model. Their data suggests SOS splicing is actually the primary pathway for removing these transposons.
And the famous spliceosome,
That's just the backup plan in this context.
it just completely flips the script on how we think about genome plasticity. Our cells have a shadow editor.
And that opens up some wild therapeutic possibilities. I mean, if we can learn to hijack that suite to maybe modulate AKP17A or RTCB, we might be able to correct genetic errors that the standard machinery can't even see.
So let's just pull this all together for a quick recap.
Go for it.
First, we have a totally new non-spliceosome pathway that's specifically designed to rescue genes from these transposon attacks.
Second, it relies on a conserved three-protein surgical team, AKP17A, CAP, or RTCB, that recognizes the physical shape of the error, not the sequence.
And third, this isn't a fluke. It's an ancient defense mechanism that redefines the spliceosome as a backup player in this specific fight.
That sums it up. Though we should add one small caveat, a limitation. We still don't know how efficient this system is across different transposon types or in different tissues.
Right. It might not work equally well everywhere. A puzzle for the next paper, I guess.
Mm-hmm.
But that leaves us with a final thought to chew on. If our cells have been secretly editing out so-called junk DNA from our mRNA transcripts this whole time, what other errors in our genome are actually being fixed before we ever see the consequences?
A very good question.
Thanks for listening to The Deep Dive.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. no paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.